Welcome to the Wheats on Your Mind podcast. My name is Aaron Harries. Wheats on Your Mind is brought to you by the Kansas Wheat Commission and Kansas Association of Wheat Growers. On this episode of the podcast, our guests are Chris and Brandi Miller with Tritica Biosciences. Dr. Chris Miller leads Tritica Biosciences as founder and chief scientific officer, where he leverages his expertise in grain science and biochemistry to develop innovative grain-based solutions. Since founding the company in 2016, he has focused on transforming traditional grain applications through biotechnology. He holds 3 degrees from Kansas State University, a PhD in biochemistry and molecular biophysics, a master's in grain science, and a bachelor's in milling science and management. Brandi Miller serves as CEO of Tritica Biosciences, where she leads strategic operations and business development initiatives. Brandi holds a master's degree in adult, occupational, and continuing education, and a bachelor's degree in bakery science and management from Kansas State University. So Tritica Biosciences is a precision biotechnology startup pioneering wheat germ-powered platform for cell-free protein synthesis. And we will explain all that in this podcast. As you might imagine, Chris and Brandi are related. A husband and wife team, good friends of mine. Thank you guys for being on the podcast.
Yeah, happy to be here.
So excited to be here.
I think your story is great. You are true entrepreneurs, and I've known you both for many years doing different things, wearing different hats. And Chris, in fact, worked here at the Kansas Wheat Innovation Center for a while for Heartland Plant Innovations. Brandi was located here at the Kansas Wheat Innovation Center for a number of years with the Kansas Co-op Council. And a few years ago, they created a company called Tritica Biosciences and doing some just fascinating work. Start off and tell us why you created Tritica Biosciences.
Oh my goodness, that's always a challenging question to answer. And really, Chris will probably just tell you why not. Why not just start this business? He has such a unique background in milling science and biochemistry that he really saw an opportunity to do something that nobody else was doing anywhere, really.
So you have facilities over by Wamego, St. George, actually, just east of Manhattan here, and that is also built from the ground up. So tell us about the facility you have, Chris.
Yeah, pretty classic startup tale. When we decided to start the company, we sold a house and we built a barn house out in rural Wamego and put a 10 by 10 laboratory and office and 100 square feet. And yeah, that was kind of the origin story. And we were able to land a couple of pivotal R&D contracts early on in the company and outgrew our space. We renovated our garage into 1,000 square feet of space. And then we kicked our family out of the house and renovated the rest of the house into another 1,000 square feet of space, ran out of that. And yeah, it's just kind of the story, how the story goes. We broke ground on another 10,000 square foot building
A couple years
ago. And I'm running out of space, so.
Well, that's always a good sign that you're outgrowing things. So
Yeah.
that does take a leap of faith. And, you know, you are raising 2 kids at the same time doing this. And Brandi, how long ago did you stop working for the Co-op Council and take the full-time leap into Tritica?
So I left the council earlier this year in January 2025. And the reason why we had decided the timing was right is because we found out last year that we were recipients of an ARPA-H project. And so it just felt like the appropriate time to step away. It was hard to leave the Co-op Council. I had a great career and really enjoyed the people I was working with. But it was at that time just Chris and our one full-time scientist, Tritica Biosciences, and that's a lot of administrative oversight required to manage a project like this. And so we just felt like this is the time to do it. It's time to go ahead and make the big final leap. And you know, I've been following Chris around his entire professional career, and so the joke is I hope this is my last stop.
Well,
Yeah, it was a very natural move for us. I mean, like Brandi kind of jokes a little bit, but we've literally worked together at almost every career stop along the way. She was a bakery science undergraduate degree. I had a milling science degree. We met at K-State. We both worked at Quaker Oats after college in Cedar Rapids. So we've just our whole lives together, we've been comfortable and used to working together. And so I like any company, your leadership team is built on trust, and I don't, there's nobody I trust more than her. So it was a perfect fit for us.
Well, and you're both really smart people, and I've seen the facility. Don't have the advantage of doing that on the podcast, but it's very impressive. Before we jump into the current project that you're working on, Chris, we should probably just kind of quickly review some of the work you were doing here at Heartland Plant Innovations, and your milling science background plays heavily into what we're going to talk about here, but your work that was being funded by the wheat checkoff here at Heartland Plant Innovations had to do with celiac disease, right?
Yeah, absolutely. And I still kind of have some loss over not being able to complete that project. And in the end, it was a fascinating project looking at the wheat origins of celiac disease and funded by the Kansas Wheat Commission. And honestly, if you look back, and this is, before Tritica Biosciences, but, that was the first step of, leaving the university and getting into the world of research and wheat-based protein biochemistry. And so I kind of have to, tip my hat to the Wheat Commission for giving me that first leap, out of the university. So, and
that work is continuing on. So through Dr. Edward Ockenoff at K-State, what you started is being built upon and having some success on that. So what is ARPA-H? I think people are familiar maybe with DARPA. That's the Defense Department research. What is ARPA-H?
Yeah, ARPA is a newer agency, and it's basically the sister agency to DARPA. I mean, it was formed by Congress as a way to utilize technology and innovation in very rapid form. And so they do project calls that are very specific to an issue or a problem that they're seeing in the healthcare space. And we have very firm, tight deliverables that the agency reviews on a monthly basis. And so, yeah, it's been a ride for sure, but ARPA-H has been a great agency to work with. They have fantastic staff, and we're not the lead PI. We have a lead PI on the project, Ginkgo Bioworks, which is Boston-based, and then there's a few other organizations that are also included in the grant or the project as well. But yeah, so that's a little bit about ARPA.
Well, let's get into the project. I continually been doing this for 19 years and always amazed by what I learned about wheat and what it can do. And this is just a whole nother level. In a nutshell, well, this really started with the milling process, right, Chris? Because the basis of your project, the matter that you're working with is wheat germ. And so there's some challenges with wheat germ stability, shelf stability, and how you separate it from the wheat kernel itself. So what was novel about that?
I think the first thing was to describe the difference between wheat germ, which you might think of as a byproduct from the flour milling process, and what our process makes is purified wheat embryo. So it is wheat germ, but it's not like the commercial flour mill product. So that in a flour milling process, the wheat germ is flaked, and it becomes an animal feed, or a certain portion of it is human food. But it, like you mentioned, it starts to decay, and it's high in lipids, and it can start to oxidize and go rancid pretty quickly after the milling process, where our process basically dislodges the embryo completely intact, and it's still living, maintains its dormancy, just like the seed is a dormant seed. So our embryo is still living and dormant, and so it doesn't have any kind of decay, and so we can keep it shelf stable at room temperature, maybe indefinitely. And that's kind of the key to Tritica Biosciences process, is that we can process and produce and accumulate large quantities of this highly, highly purified wheat embryo.
And like we mentioned when you came out to visit the facility, what makes that piece so unique is that we didn't invent cell-free protein synthesis using wheat germ. There was actually work done by USDA back in the 40s and 50s utilizing wheat germ, but as wheat growers and people very knowledgeable about wheat, you understand the kernel and the aspects of the kernel, and the former process or protocol to remove the embryo, like Chris was mentioning, was actually using a scalpel. And so what makes our process so unique is that we can scale it because we can process significantly large amounts of embryo to be utilized in cell-free protein synthesis, and that's just never been done before.
Yeah, so wheat germ is actually one of nature's most healthy foods. It's got a lot of good parts in it, and you can buy it in the grocery store on the shelf, but it's not very shelf stable. But you're using it for a whole different purpose, and instead of keeping people in suspense, let's talk about what cell-free protein synthesis is and what you can use wheat germ for in that capacity.
Something that we have found to be very helpful when we're talking about this process, because it's pretty scientific and it's pretty deep, is Chris has a great analogy that he uses with regards to ethanol to kind of talk through the process, because most people are not familiar, even scientists are not always familiar with cell-free protein synthesis. So I was just kind of setting you up for success there.
Okay, thanks.
Good teamwork.
Right. We are in this area of bioprocessing called cell-free protein synthesis. And you can imagine, I think most people know that inside every living cell is the cellular machinery that makes you living. And it's basically every cell has the ability, it has ribosomes and enzymes that would take your DNA, convert your DNA into RNA into protein. So the DNA in every cell is the instructions that your body uses to make every single protein that makes you, you. And so in cell-free protein synthesis, we don't need a living cell. And so the machines will work outside of the living cell, believe it or not. And like Brandi said, we didn't invent that. But what we can do is, because our wheat germ is the way that it is, we can extract those cellular machines and keep them intact and still working. And so then at that point, we can add instructions or DNA for really anything. It's not a plant anymore at that point. It's just a set of cellular machines. And so we can put in DNA for human insulin, and the machines will pick up that instruction set and make it immediately.
They're little factories.
Little factories, yeah.
Wow.
And so, yeah. And I think that kind of is a call back to my milling science degree, and it's a factory, it's a process, it's a set of machines put together in a certain way to make a product from an ingredient. And that's probably the way I see things more often than not.
So in a nutshell, you're using wheat germ to create insulin is one of the applications that extends way beyond that. So Brandi, when we were visiting you the other week, you showed us a package of wheat germ, right? And for people who don't know what it looks like, it's just, I don't know, it's itty bitty pebbles inside a bag, and they're shelf stable, and you can send those anywhere, right?
Right, absolutely. I mean, that's what makes wheat germ so special, is because it is shelf stable. I mean, it's meant to feed a grain as it grows into a plant, and so that's what it's made to do, is just to be dormant until you need it. So
you can take these packages of wheat germ, which is kind of the product there, and ship it around the world, and at that location. So if Doctor had a package of this specialized wheat germ, what's the process then to get to the end result, Chris?
Well, we actually have one more step before we will send the raw germ out. So we actually create a lysate or a slurry that we extract the basic workings that Chris was talking about. So the machinery, that's what we extract, and that is what we would send. Because that's what you add the RNA or the DNA to. Yeah,
that's kind of the secret sauce in our process, is our ability to make this extract and maintain the productivity of those machines.
How do you make that? Is it a complex process, or is it fairly easy?
Well, there's parts of it that I can't totally talk about, but in general, you could imagine just like any extraction process is just like making a smoothie, and you basically just gonna grind it up in a special smoothie recipe, so to speak, and from that smoothie, then we can make our extract of the cellular machines. So
that extract is extracting essentially those workings of the cells in the wheat germ.
Yeah, and that's why the wheat germ is so. I mean, the wheat germ is critically important to the process, just because of what it is. Like Brandi said, it's the seed goes in the soil, and that embryo has to have every resource that it's going to take to get through the soil until it can get a leaf up and start photosynthesis. And so it's packed. I mean, it's just a very densely concentrated abundance of these machines. Not you couldn't go take leaves or grass clippings or some other vegetative tissue and do what we do. It's really specific to the embryo.
So what are the potential applications of something like this?
We don't even know fully, to be honest. I mean, we are just getting started. Our big focus right now with the ARPA-H project is to focus on the pharmaceutical deliverables. But as Chris mentioned, we've had success with enzymes. We've had success with human DNA, plant. I mean, we really think it's endless, honestly. It's just a matter of optimization because every protein comes with some of its own challenges. And so figuring out how to make it work within our system is really our goal.
So, for example, insulin doesn't travel well, right? So if you sent some of this extract or the germ and they created the extract to Africa, are they able to, in a sense, manufacture insulin there on site?
So what we're actually working on is pretty cool, but the second part of a seed being a seed is that it also likes to be dry. It has no problem being completely desiccated in a dry state. And so we found that once we extract these cellular machines, they're pretty happy being dry as well. And so we can dry that down into a powder. Mm-hmm. And the whole concoction is already, blended into this powder. And so really we can have a just add water bioprocess where you literally just add water and DNA for your target and you can start production immediately. It's not like a fermentation process is what we would normally see for bioproduction or bioprocessing, and this is what Brandi's talking about. Ethanol is a bioprocess where you use yeast to produce the ethanol, but you have to grow the yeast up in a huge, bioreactor. And yeah, in this case, there's no delay. You're not growing a living cell. You don't even have to keep the living cell alive because there's no living cell. So
there's a lot of advantages of utilizing the cell-free process because it does eliminate time is one of them, and the other is just the scale of what we're able to do with the space that we have versus needing massive bioreactors to
the
do
equipment, the efficiency
right.
is just much less cost.
Right. And the beautiful thing about germ is that the byproduct is a human consumable product. We could just take it and make flour out of it. Yeah, and so on the front end of the process, on our platform itself, it is also sustainable and has an application where we're not just wasting it.
Yeah, a lot of fermentation processes. You have a lot of wastewater, management that you have to deal with, and waste products that just have no value. They're actually be a cost, and so. Yeah, our process is totally food safe, and we don't add any kind of chemicals or anything to our process. And so the wheat, the remaining 98% of the seed is still perfectly suitable for the flour milling process. So it could, to some point when we scale to a level where we need a flour mill to get rid of our quote unquote waste, that'll be pretty sweet. But it'll go directly into a flour mill and yeah, it's completely 100% waste-free.
So I don't think it's any secret that this is a $29 million 2-year contract. 4 partners involved total?
We actually added a 5th just recently, but 4 initial partners that are focusing predominantly on the science, and our latest addition is kind of helping more with the economic analysis components of it. But yes, yep.
So what are the other partners' role in this project?
Their focus is really on more of the actual protein synthesis using our platform. So they are healthcare companies that are focused on utilizing the platform to do conversions into these actual drug deliverables, where we're essentially at the very beginning of the process. And so we provide this platform for them, and then they utilize the platform to manufacture the drugs.
Does the type of wheat matter?
Oh, that's a really good question, Aaron. And because you understand this business, you ask that question, where it's interesting when we've had scientists come out to visit, they don't fully grasp the fact that not only does there's more than one class of wheat, obviously, and you also recognize and understand, and I'm sure a lot of people that listen to this will also understand there's a lot of different varieties too. Mm-hmm. And we just have started really tapping into understanding what that looks like. What we're finding is a growing region, maybe the most important, but again, this is still pretty novel, and so nobody else has looked at cell-free protein synthesis using wheat germ at this level, and so we got some work to do, but we're seeing some.
So the short answer is
yes.
Yes, it
absolutely.
And we've been working with Bryson, KWA to get Kansas grown wheat, hard red winter wheat, and really need to tap into Alan's brain power about wheat varieties and what might be out there that we're not seeing. But the winter durum, we've tested that through the facility and about 6 other Kansas hard red winter wheat varieties out of Alan's program, and then Brett Carver sent us some of the purple wheat that actually came out of Alan's program, and I think he shared with Brett, and it's working. So it's really interesting.
So I'm curious, do the applications extend beyond pharmaceuticals? I mean,
sure.
What do bioreactors typically produce?
I mean, if you think about proteins just in general, a lot of things are manufactured by protein. As a person who enjoys cosmetics, there's a lot of protein applications in cosmetics. We haven't even started exploring some of the pet food applications, which I think those could be really exciting. I do think, though, that taking a step back, we really have to understand and make sure that it's still cost effective. I mean, we've made chicken eggs, and it was really expensive, and it's still cheaper to just grow chicken. So finding applications that are cost effective is really where there's value add and the cost makes sense.
So even with the price of egg at $5,
even with the price of egg, even at $10 a dozen, it's still cheaper to just buy a couple chickens.
That was actually the very first thing that we thought we might do at Tritica with our platform was all these companies trying to make egg replacers out of peas and beans and all kinds of plant-based things to make these eggs. And I was like, well, let's just make eggs. Put a lot of energy into beaks and feathers when you just want the egg anyway. And so we went after it. And we actually did make the eggs, not a full egg, just egg whites, because it's pretty much just one protein. But yeah, in the end, turns out it was cheaper just to grow chicken. But
so what comes first, the chicken or the egg? You're just like, what chicken?
Right. Just the egg.
Yeah. Okay. Well, fascinating stuff. So what would this business look like scalable? I mean, you talked about, so you're buying wheat, you're milling it a special milling way to knock that germ off. The byproduct just goes back into a milling system. So, who would be doing that? Would it be a traditional flour mill in your mind that would be doing that kind of process? And how have you thought that far down the road?
I don't know for sure that our process could actually be in a flour mill. I think, after getting this far down the road and realizing that there is a certain level of cleanliness that you have to have, even on a food grade product, it's like going to be feeding and potentially a pharmaceutical kind of has to be done. Not totally in a pharma like level of cleanliness, but it's a little bit more than a flour mill. And so probably we would truck. It might be adjacent to a flour mill, but I think that's the most logical outlet for our wheat, our spent wheat, would be a human food product. I mean, yeah, I just see it being that way.
Well, how cool would it be to think about using Kansas wheat to manufacture insulin? I mean, that's a cool thought process. If we had a farmer that could produce a particular variety of wheat that would be utilized for just insulin, or we plug insulin in because it's something that we've been working on with our partners and because a lot of people are familiar with it. But there's a lot of other protein-based drugs that could be manufactured using our platform.
Yeah, and it doesn't have to just be drugs or proteins either. So most of the small molecules or sugars and things that are compounds, vitamins and things like that are in plants, I mean, those are not proteins, but they're made by proteins called enzymes. And so an enzyme is just a bioactive form of a protein that can convert one small molecule into another. So one of the other parts of the project is to make what's called APIs, which are pharmaceutical ingredients, which are small molecules. And so typically these things are made in A lot of times China and India, and they're made through chemical synthesis routes that require, toxic chemicals, environmentally hazardous, terrible in some cases. And so us and the partners have come up with a biocatalysis, so using enzymes to catalyze these molecules directly from some kind of precursor, sugar or amino acid. And so in some cases, we actually took a 20-step chemical synthesis route, which is the standard route to produce this compound, and they came up with a 3-step enzymatic conversion. It actually makes it's more efficient to produce. It's greener. It's crazy. So we're not limited to just making proteins or things that are coded by DNA. Once we understand the enzymes that can convert one sugar into another, well, then we can pretty much make anything, I think.
Yeah. Do you think it has any food industry applications?
Back to absolutely. The answer is absolutely. But back to the conversation we had just a few minutes ago, I think the biggest challenge is just making sure it's cost effective.
Mm-hmm.
That's where we would have to probably become a little bit more efficient and optimized in order for it to financially make sense. The food industry, as you know, is just, the margins are so tight. And so even though we can effectively produce probably several different components that are utilized in the food industry, it's still an expensive process.
And we probably won't even be able to compete with some really efficient fermentation processes. I mean, yeah, we'd like to say that we can just eliminate all bacterial fermentations, but the reality is that's just not true. Like, there's some current processes that are made through yeast or bacillus or some other fermentation, and we'll never beat them because they're just so dang. Like, some of the enzymes, we thought about biofuels, like maybe producing enzymes to help with biofuel production, and they can make those so cheap that they can't even afford to ship it in plastic. They just put the enzyme plant next to the biorefinery and pipe it across.
Right.
So, I mean, you just can't compete with that, I think.
Yeah. I do think it's great what you mentioned earlier, Brandi, though. Farmers saving lives through feeding people wheat, but just another application here, just another story to tell would be something awesome to do. So what happens at the end of 2 years, at the end of this 2-year contract that you have with ARPA-H?
Well, when we originally proposed the project, it was actually a 4-year commitment. And so our hope is that ARPA's been really pleased with the work that we've done, and we're hopeful they will extend it for 2 more years. It's a lot of work to be done in just 2 years time frame. And so we really are excited about, we've had Senator Marshall come to visit and just engaged Senator Moran's office, and just having legislators come out and see the really positive work that we're doing, and hope that we can extend it 2 years. The other side of the ARPA-H proposals are that there's always a commercialization mandate. And so that's the end goal, right? Is to figure out how we can kind of commercialize this and get it into the hands of people that can utilize it and manufacture many things with it. And so I don't know. I mean, candidly, I don't know today if we can do it in 2 years. So I'm really hoping that we can get an extension, but we're going to be really close. The amount of work that we have done since April is absolutely incredible. It's really been phenomenal to see how much work we've gotten done since we started this project.
How many employees are you up to now?
Well, we just had a third employee started today. And so we have 3 full-time scientists on staff, plus Chris and myself. And then we have a couple of contract employees as well that help us part-time. And we have an intern that will be staying with us through the fall. And then we're looking at potentially 1 or 2 more hires. The other beautiful thing about what we do is it doesn't take a lot of manpower. We're able to be pretty efficient with the individuals and level of skills that we have. And so we have a great team. Our people are just really incredible.
So Tritica Biosciences does other work too, some proprietary stuff I know you can't talk about, but give us an insight into maybe some other projects you've got going on.
I mean, I think the platform itself, I mean, people from the outside looking in are starting to learn more about it, learning that we exist even. We've been pretty quiet over the last few years. So just being able to produce other people's protein of interest. And so you can imagine, AI is everything. Everything in the world today is AI, it seems like. And so, an area of research is trying to predict and produce proteins that maybe don't even exist in nature. They're just completely dreamed up from a computer, from AI. And You can make a lot more protein sequences in the computer than you can actually get your hands on. And so we're helping some AI folks get their hands on the proteins that they're designing, in the computer. And so our system's really, it's made for that. And so we take the gene sequence out of the computer and we put it into real life and make a real gene out of it, real DNA, and then we just put that into our system as an ingredient, and it makes that thing. It's kind of weird.
So I've got to ask you a question, Chris. There's change in the wheat industry now. We're kind of entering the GMO era of wheat, which is long overdue, I think, and we're moving down that path. So you, as a scientist, I always like to ask, when you get down to plant DNA, I mean, it's kind of the same, isn't it?
Yeah, totally.
From plant to plant. I mean, it's the same building Legos, isn't it?
It is. Yeah. No, I'm a total supporter of moving in this direction for our industry, and our process that we do, our cell-free process, is technically a non-GMO technique. It's non-GMO, but if I had access to GMO wheat, I would make a wheat variety. It wouldn't be a good bread baking variety. It wouldn't be a feed variety or whatever it may be. It would be a protein translation machine. It would be for me, for my process. Yeah.
Yeah.
Totally.
How do you stay on the front edge of all this technology? I mean, the AI, the equipment you have in your lab is amazing, but yeah, how do you stay on the edge of that, finding what's new, what capacities you can have in that, and still doing your job at the same time?
There's a lot of sleep at night thinking about this.
That's a great question.
Yeah,
I think
scary.
We're fortunate because we had a awesome partner in the first 5 years of kind of focusing on this technology, and we've locked a lot of this IP up with patents, and so it's going to give us a little bit of headwind or leeway. I don't know. That's not the right word I want to use, but a little bit of runway, I guess, but not much, because I really think once people start seeing that this works, I think we'll have a lot more interest in competition. Right now, nobody's competing because nobody believes it's true. Mm-hmm. I mean, that's really the truth, because, you think about what the lab protocol is. It's using a scalpel. The fact that we can mass produce germ is what makes our process so unique, and nobody's been able to do that yet. And so I think that's the biggest concern.
There are other model systems for cell-free. So there are companies in the commercial space today that are doing drug manufacturing using an E. coli-based cell-free extract. So basically, we're using the embryo to extract our machine. So this company is growing up E. coli and then using those dead E. coli cells to extract the machines out of the bacteria. But it takes 1,000 liters of fermentation for E. coli to produce 1 liter of cell-free extract for the cell-free system using E. coli. So the scale there it's backwards. It just doesn't.
Well, and wheat germ sounds a lot friendlier.
It does sound friendlier, right?
Yeah.
But we could do that, with a 10-gallon, 5-gallon bucket of wheat.
Yeah. Well, I think this is a great story, not only in the science that you guys are doing, but that you're entrepreneurs that built this from the ground up and are having success. And I wish you all the best. Thanks for sharing. If people want to follow and keep following your story, I mean, how do they find out more?
Well, they can check out our website, triticabio.com, or we're on LinkedIn. It's probably where we're the most active as far as watching what we're doing. And yeah, if anybody has any questions, certainly info@triticabio.com would be a great place to reach out.
And we will have you both back when you make new discoveries and get an update on how this is going. So Chris and Brandi, thanks for joining us.
Aaron, thank you so much. Really appreciate it.
My thanks to Chris and Brandi Miller for joining me in this episode of the podcast. If you have follow-up questions for Chris or Brandi, please email us at kswheat@kswheat.com. You can find all previous episodes of the podcast at wheatsonyourmind.com. I'm Aaron Harries. Thanks for listening.